We looked at hexokinase, we looked at PFK1, we looked at FBPS1, we looked at FBPS2, PFK2, pyruvate kinase, right? Those are the main ones we looked at. And so, what you have to think is that you have to first think about the metabolic condition. Are you fed or are you starved? Then, if you are, let's do starved first. So, if you are starved, the enzymes that will release glucose will be promoted, and the enzymes that trap glucose or do something with it will be downregulated. On the other hand, if you are fed, then you want to promote processes that will use glucose to do something, either do glycolysis or store it as glycogen. So, that is the overview of how you should remember. Under starved states, phosphorylation happens, right, with the kinases, and phosphorylation can either make enzymes active or inactive depending on the context. Some enzymes will be activated by phosphorylation, some enzymes will be inactivated by phosphorylation. The enzymes that are consuming glucose will be where, in those cases, phosphorylation will inactivate them. For example, we saw last class. Pyruvate kinase, also PFK2. So, those enzymes will be inhibited by phosphorylation because they do something with glucose. So, under starved condition, we do not want glucose to be utilized, we want glucose to be released into the blood so that other tissues and brain can use it. And if you are a fed condition, then you want to promote processes which will do something with glucose. For example, PFK2 is promoted, right, by phosphatase, which is activated by insulin. Are you looking at fed state or are you looking at starved state? And then decide which enzyme is doing what. If it is releasing glucose into the blood, then under starved state those enzymes will be promoted, and in the fed state the enzymes that are trapping glucose will be promoted.